Hello, I'm Manariza, and welcome back to Lectures on Explicit Memory. Well, as I explained in the previous lecture, we are still at the section 3 of those lectures on explicit memory. And again, as I told you in the previous lecture, I dedicated the whole section 3 of the lectures on explicit memory just to microcircuits, an introduction to microcircuits, and hippocampal microcircuits, okay? So, this is the outline of the whole explicit memory lectures. You know, I talked about hippocampus and LTP, but the third section is just about brain microcircuits and hippocampal microcircuits. And this is the outline for these lectures, the third section. And in the previous lecture, I introduced microcircuits, I defined microcircuits, I talked about iconic synaptic patterns, and I talked a little bit about the historical background of microcircuits. And now, in this particular lecture, I'm going to talk about microcircuit studies. And, yeah, and in the future, in the next lecture, I'm going to talk about cell assemblies and dynamic aspects of microcircuits, and finally, there's going to be a separate lecture on hippocampal microcircuits. But for this lecture, I'm just going to talk about the microcircuit studies. Well, I don't want to dive deep into the history of microcircuits. I just want to introduce two of the most important pioneers of microcircuit studies, two of the giants of microcircuit studies. One of them is Rodney J. Douglas, who for the very first time introduced the idea of a canonical microcircuit. Well, a canonical microcircuit is just a basic, simplified, or a generalized way to demonstrate a microcircuit. And for instance, this is a proposed model for the microcircuits of the mammalian visual cortex, and it's a canonical microcircuit because it is simplified and very general. It shows us a general structure or the general map of connectivity in that very complicated microcircuit. We all know the visual cortex is so complicated, but you can see that this figure is very general and simplified. And I told you that canonical microcircuits are basically, the concept of canonical microcircuits is introduced by Rodney J. Douglas. And so let me explain this figure. You know, in the mammalian visual cortex, in the proposed model of the canonical microcircuit, in the mammalian visual cortex, we have three populations of neurons. We have two populations of neurons are excitatory neurons, and we have an inhibitory neurons, a population of inhibitory neurons, or here represented by these GABAergic cells. And one of those excitatory populations of excitatory neurons consists of those neurons at the principal neurons or pyramidal neurons at layers 2 and 3, superficial layers of the cortex. And then spiny stellate cells, because some of their properties resemble the properties of pyramidal cells at layers 2 and 3, they are also included in this population of neurons. And then we have another population of excitatory neurons that are at deeper layers, layers 5 and 6. And as I told you, we have an inhibitory populations of neurons, or populations of inhibitory neurons. We have a population of inhibitory neurons, which are also called GABAergic cells. And then there are, connections between these populations of neurons. We have several, we can identify those iconic neural circuits. For example, I told you that this is a population of excitatory neurons or pyramidal neurons. This is another population of pyramidal neurons or excitatory neurons. So this synapse is actually a feed-forward connection, a feed-forward excitation, okay? From these excitatory neurons to these excitatory neurons, from superficial ones and to deeper layer ones. And then we have this kind of connection, which is recurrent excitation or feedback excitation, because these neurons actually connect to themselves. And they also activate these GABAergic cells. Well, these triangles, they represent synapses, but those open triangles or white triangles are excitatory synapses, and those black ones are, or filled ones, are inhibitory synapses. You can see that these, the population of GABAergic cells make connections with themselves, recurrent or feedback inhibition. They also inhibit other, both of these populations of neurons, those at superficial layers or at deeper layers. And the thickness of lines represent the strength of connection. For instance, you can see that this line from GABAergic cells to deeper layer pyramidal cells is thicker than this line. It means that this inhibitory connection is stronger than this one. Okay, and then there we have thalamocortical or thalamic input to all of these three populations of neurons. So you can see that canonical microcircuits are very good, although they have those two problems that I introduced in the previous lecture, that they lack structural complexity or the functional dynamics of these synapses. But they are still a good way to give us a very general view of a very, very complicated microcircuit. Because, for example, visual cortex the microcircuitry in visual cortex, basically in cortex, is very complicated and elaborate and intricate. But, a canonical microcircuit can give us a general and a very simple to understand, a simple map of all of those connections, of the most important connections in a single microcircuit. Well, other microcircuits are also other canonical microcircuits are also introduced by another giant and another pioneer of microcircuit studies, Professor Gordon Shepherd, the author of the book Handbook of Brain Microcircuits, also the author of the book. He also wrote the book called Synaptic Organization. Well, Professor Gordon Shepherd proposed a model for canonical microcircuits for 3-layer simple cortices and 6-layer neocortex. We have, according to Professor Gordon Shepherd, we have two types of cortices. We have those 3-layer ones and those 6-layer ones. For example, an example of a 3-layer simple cortex is just the olfactory, is olfactory cortex. Okay, so for example, here is a canonical microcircuit proposed by Professor Gordon Shepherd. It's a canonical microcircuit for the olfactory general or hippocampal general cortex, and you can see that we have 3 layers, and of course we have subdivisions of layers. For example, layer 1 has, we have 1A and 1B. Anyway, we have 3 general layers. And then we have primary afferents, and we have, those iconic neural circuits that I introduced in the previous lecture are already identified here. For instance, we have feed-forward inhibition. You can see that this is an axon of an excitatory neuron activating this inhibitory interneuron, and this in turn inhibits this pyramidal neuron or excitatory neuron. And then we have recurrent excitation. You can see one of the axon collaterals of this pyramidal neuron goes up and activates itself. Or we have lateral inhibition. That when this inhibitory neuron is activated, you can see that it inhibits these two lateral neurons. So we have those, iconic neural circuits in this canonical basic circuit representing a microcircuit for a 3-layer cortex. And then we have a microcircuit, a canonical microcircuit suggested for the neocortex, which has 6 layers, more layers, and it's way more complicated, but we still have those iconic neural circuits like the lateral inhibition, recurrent excitation, lateral inhibition, feed-forward inhibition, and surprisingly, remember those arrows in Cajol's drawings? I told you that Cajol drew those arrows in his drawings to represent the flow of information or the direction of information processing. We still use those arrows here. We have those here. Information goes out from primary afferents, and then information goes down via these cortical connections. Wonderful. So these are, the very first serious studies on microcircuits. Cajol didn't study the microcircuits very seriously, but Professor Gordon Shepherd, he's a pioneer of microcircuit studies. Although his he mostly worked on olfactory system and the microcircuits of the olfactory cortex, but he actually started the serious studies on microcircuits. Okay. So, in the last lecture, and I also mentioned in this lecture, that these iconic schematic or canonical microcircuits or basic circuits, they cannot show us the true complexity of the true structural complexity of real microcircuits in the brain. And these synapses that they actually show or demonstrate are static. They're not dynamic. Okay, they don't change. But we know that real synapses in real microcircuits, they change over time. They're plastic. They change. So I want to introduce two of the biggest projects in the history of neuroscience that are trying to tackle those problems, that are trying to understand the structural complexity and model that structural complexity of the brain and the microcircuits or connections of the brain, actually. And in another lecture, I'm going to talk about how scientists are trying to understand the dynamic aspects of microcircuits. But in this lecture, I'm just going to introduce those two lectures that are trying to understand and model the structural complexity of real microcircuits. Well, one of those projects is called Blue Brain Project. Of course, you have heard about it. It is started by Professor Henry Markram. Professor Henry Markram, I love him. He's a wonderful scientist, a neuroscientist. He's a visionary neuroscientist. And he started the Blue Brain Project. And, well, this is a picture summary of one of the papers that they published, 2015, I guess. And, well, this picture summary summarizes the steps of the Blue Brain Project in order to, the main purpose of the Blue Brain Project right now, at this time, is to reconstruct the a volume of neocortical microcircuits. Okay, they're trying to model a volume of neocortical microcircuits. It's a very difficult job, but they're trying to do that, and to some extent they succeeded in doing it. So, what I love about the Blue Brain Project and the mind behind it, Professor Henry Markram, is that in the Blue Brain Project they are trying to, model the the neocortical microcircuits as real as possible. They're trying to make their models very close to biological microcircuits or real microcircuits. And the way they do that, the very first step in reconstructing a microcircuit in order to model it is to gather experimental data, okay? So they first acquire experimental data from the real brain, okay? This is a mouse brain or the brain of a rat, I'm not sure. But they actually use real brain to get their data. And what they do is that first they try to understand the firing patterns or the electrophysiological properties of individual cell types in the neocortex or neocortical microcircuits. And then they try to understand the very complicated morphology or the shape of individual cell types of neurons. And after that, they try to reconstruct the cellular and synaptic anatomy and physiology of those neurons and microcircuits in the real brain. This is very important because they not only they try to reconstruct the anatomy, but they also try to imitate and model the physiology of cells, of neurons and their synapses. Of course, they do that in silico. It means that they do that by computational models in these supercomputers. And this is one of those models that they made. You can see that these neurons are morphologically correct. Their morphologies are similar to the morphologies of real neurons in real brain microcircuits. And of course, they have they imitate the real firing patterns or the firing properties of those neurons in real microcircuit. And of course, they use their models for some simulations, and they were really successful in predicting the in vivo state by using their simulations, and they did very successful simulations. I learned from this project and other scientists in the field of microcircuit studies. Sorry. I learned from Professor Henry Markram and other scientists, and of course from the book by Professor Gordon Shepherd, that one of the very first steps in any microcircuit studies, in trying to understand a microcircuit, is to identify the types of neurons that are that comprise a single microcircuit. It means to distinguish different cell types. And neuroscientists, like Professor Henry Markram and his team, they use three properties of neurons or cell types of neurons to distinguish and to put them into different categories or different types of different cell types. First, they use the morphology or the shape of a neuron. They have different techniques to detect the 3D structure or the 3D morphology of neurons. But it works most of the time. But sometimes it doesn't, because sometimes, although different cell types may have different morphologies, but sometimes there may be some overlaps between the morphologies of different cell types. Another feature of a neuron or a cell type or a specific cell type they use to distinguish a neuron from others is the electrophysiological properties of that specific neuron or the firing pattern. I'm going to introduce different firing patterns of neurons, but again, we're going to see that sometimes the firing patterns may overlap, or sometimes we have one neuron with different firing patterns. Wonderful. But that increases the difficulty of identifying cell types. And finally, they can use the molecular profile of a single neuron in order to distinguish it from other neurons or other types of neurons. Scientists found out that some neurons or some specific cell types of neurons, they express very specific proteins. For instance, there is a group of interneurons that express parvalbumin, which is a protein, and they can use that parvalbumin, the expression of that parvalbumin, to distinguish those interneurons from other interneurons, because there are some other interneurons that do not express parvalbumin, but for example they express cholecystokinin or CCK, okay? And they use these molecular markers of individual cell types to distinguish neurons. So let's see what scientists did in the Blue Brain project to identify cell types. First, they worked on the morphological diversity of neurons, and they identified they tried to understand the morphologies, they tried to reconstruct the morphologies of neurons that are part of the neocortical microcircuit. And you can see that these blue ones, okay, these are interneurons or inhibitory neurons, and these red ones are pyramidal cells or excitatory ones. And you can see that the different types of interneurons and different types of principal neurons or pyramidal neurons, they have different morphologies, okay? Even sometimes the morphology of the same cell type may differ layer by layer. For example, the morphology of a nested basket cell in layer 2 or 3 is different from the morphology of the same type of neuron, nested basket cell, but at layer 4, for example. So they have different, different morphologies. Basket cells, bipolar cells, chandelier cells. These are different morphologies, different names given to different morphologies of these inhibitory neurons. And then we have principal cell pyramidal neurons or spiny stellate cells, and which are also excitatory, and different morphologies. But I told you that another feature of a neuron that scientists can use in order to distinguish a cell type of a neuron from others is by using the firing patterns or the firing properties of a single neuron. Well, different neurons have different firing properties, and I'm going to introduce some of those common firing properties of different neurons. For example, when we actually inject a step current, whether a low step current or a high step current, we have a neuron that when we inject that step current, it fires regularly in a tonic way. It fires regularly and with the same inter-spike intervals, nothing crazy, nothing unusual, just a regular kind of firing. But when we inject the same kind of current to another neuron, first of all, we have a very rapid sequence like of action potentials. Okay, we have a sequence of action potentials that happen that are fired rapidly, and then there's a rapid transition to that tonic one. Okay, to that regular firing again, and this is called burst tonic. We have an initial burst, and then we have a tonic, and after that, and then we have delay tonic. When we inject that current, we first have a delay at the beginning, okay, and then we go on and the neuron fires tonically or regularly. So these are three different types of firing patterns that can be seen in three different types of neurons, actually. It's not finished, of course not. We have another type of firing pattern called adapting. In adapting firing pattern, or in the phenomenon called adaptation, what happens is that first we have, when we inject that step current, we have some action potentials, and the inter-spike interval, the interval between two action potentials or two spikes, is shorter at the beginning, but later it gets longer at the end. Toward the end it gets longer. And this phenomenon is called adapting or adaptation. And so we have adaptation or adapting. Then we can have burst adapting. Like burst tonic, we first have a burst of action potentials and then adaptation at the end. And then we have delay adapting. We have a delay at the beginning and then regular firing, but at the end we have adaptation. Adaptation is just all about that longer inter-spike interval toward the end of action potentials, or toward the end of firing of a neuron. And then we have these weird and bizarre firing patterns. For example, there is a regular burst, then we have a burst, and then a pause, then a burst, and then a pause. And you may think that these, we have a pause after each burst because we stopped the current. No, the step current is kept constant. But then we have pause, pause, pause. We have burst and pause and burst and pause. So we have, this is called regular burst. And then we have initial burst. Okay, we have a burst, and then maybe we may have tonic ones or very few action potentials after that initial burst. And then we have transient ones, that we have some action potentials some of the time, not all the time. So these are common firing patterns of cortical neurons, of course. And, those firing patterns were also considered in the same project. This is yet another figure from the same paper published by Professor Henry Markram in 2015. In 2015, and his team, of course. And they use different terminology. For example, they use continuous for regular kind of thing, accommodating for, I told you that we have tonic, burst, and delay. They have burst, delay, and continuous. Okay. Then we have they use the word accommodating, I think, for adaptation or adapting. And there is stuttering ones, there is irregular ones, burst irregular. First we have a burst and then irregular firing pattern. And different firing patterns, but this is not the case. This is not important here. What's important is the part B of this figure. This is a layer 2 or 3 nested basket cell. And basket cells are a type of inhibitory cells. They're interneurons. And the reason they're called basket cells is because their axons actually form a basket around the postsynaptic target, around the cell body of the postsynaptic target. Anyway, so this is a cell type, a single cell type, the Nesket basket cell. But you can see that it may, it shows, it may have different firing patterns in different occasions. Under different conditions, it may show different firing patterns. I mean, 30% of the time, for example, it has delayed non-accommodating firing pattern. But 5% of the times, it may have a burst accommodating firing pattern or a continuous irregular firing pattern. What is really important, this is very interesting and very weird. A single cell type can have different firing patterns under different conditions. And so that increases the level of complexity and the difficulty, of course, in microcircuit studies. Because I told you that scientists try to use firing patterns to distinguish a cell type. But when a single cell type may have different firing patterns, that increases the difficulty of a problem that we are trying to solve. This is very important. Just wanted to mention that. Okay. Well, I forgot to change the title of this slide. Well, cell assemblies, yes, of course. I'm going to talk about them in the next lecture. But cell assemblies are somehow part of the microcircuit. Microcircuit is a bigger structure or construction than cell assemblies. But this is a short clip from the Blue Brain project. I just want you to see that because it is amazing, okay? Let's just see that together. This is a neuron that its morphology is reconstructed and is modeled, okay, based on real neurons in Silico by the team of Professor Henry Markham. And it also displays the real firing patterns of the firing patterns of the real neuron, okay, in the real brain. You can see that they tried really hard to model the exact morphology of a neuron. And then they add more neurons. And they also, I told you that they tried to reconstruct the cellular and synaptic anatomy and physiology. They also model the synaptic connections between these neurons. It's wonderful. It is very wonderful. And then all of these neurons can fire according to their innate or default firing pattern somehow. This is beautiful. I mean, this level of complexity in modeling is mind-blowing, Yeah. This is that volume of neocortex that they try to model its microcircuits, okay? That is the volume of this is a reconstruction of a piece of neocortex. The microcircuits of a piece or volume of neocortex. Okay, I think that's enough. So that was about the Human Blue Brain project. They tried to reconstruct and model a piece of microcircuitry of the neocortex, and they're doing a great job. They're trying to model both the anatomy and physiology of the microcircuits. But there's another project going on right now called Connectomics. Yes. Well, this project was started by Professor Jeff Lichtman, another visionary scientist and neuroscientist from Harvard University, my favorite university. So let me just go on and explain what is happening in this project. Well, this is again a picture summary of the paper published by Professor Lichtman and his team. What they do is very interesting. First of all, they prepare some samples for electron microscopy. But because their sectioning, okay, for sample preparation is done automatically, they put those. I mean, this is the device. This is ATUM cutting and collecting device. This device automatically cuts and collects the slides or samples or slices. And because this machine does it automatically, it puts these slices or samples on a tape, okay? And later, people in the project and Professor Lichtman's team, they actually cut that tape and put it on a kind of wafer or a plate. I'm going to show you the picture. But anyway, first they prepare some samples. They prepare it for sectioning and cutting and collecting. And then what they do is that they do scanning electron microscopy. And then they do saturated annotation, really sorry. After scanning electron microscopy, they do saturated annotation. By saturated annotation, what they do is that they assign a color to those structures that they can identify. Okay, each one of these structures can be, for example, a neuron or a section of a dendritic shaft, for example, or a section of an axonal terminal, and they assign a color, and they call that task saturated annotation. After that, they try to move, and they stack those annotated sections on top of each other, and they try to follow those structures with the same color through the frames, or through the sections. Of course, they do that by computer softwares. And that computer software automatically reconstruct a 3D structure of cortical connectivity, of wires of the brain, of synapses and axons and dendrites. There is a movie, actually, I'm going to actually show you, because it's very hard to explain what they do. I think it's good to. This is Bobby Kasthuri. I'm not sure about his last name, how to pronounce it correctly, but he's one of the leading scientists in the project of Connectomics. These are the strips or parts of that tape that I told you in the previous slide. I think this is a microscope, not that cutting, not that machine that prepares the samples, but that machine, that cutting machine, automatically collects and prepares some slices, we can say, and collect them on a piece of tape. And these are the strips of that tape, the parts of that tape on this wafer, on this disc, and this plate. And each one of these things here are slices of a real brain. Okay, I think it is the brain of a mouse. And then they do a very high resolution electron microscopy. Okay, then they assign a single color to a same structure, and when they move through those slides or sections, they actually get a 3D structure, okay? And this is the saturated annotation of those electron micrographs. It's wonderful. And finally, they get these very beautiful and very intricate, very complicated maps of cortical connectivity. What I wanted to tell you. Oh, stacking those slides or those electron micrographs on top of each other and moving through them, it's just like moving through the frames of a movie. When you move frame by frame, you actually move through time. But when you move through the slides, okay, that you stack them on top of each other, you move through space. And that's how that computer software is capable of reconstructing the 3D structure of cortical connectivity or wires of axons and dendrites. So, real structural complexity. This is a figure from the Blue Brain project. You can see that this is the in silico diversity of inhibitory morphological types. These are the morphologies of inhibitory neurons that they try to reconstruct in the Blue Brain project. You can see that Henry Markram and his team are trying to understand the true structural complexity of microcircuits. Something that you cannot see in canonical microcircuits. This is a beautiful picture or figure from the Connectomics project by Professor Jeff Lichtman. It's so wonderful. These numbered structures, these are individual synapses. And in the Connectomics, in the project Connectomics, scientists are trying to map the cortical connectivity at the resolution of individual synapses. This is wonderful. You can see that this is one we have one synapse here, another one here. I think this is a dendritic shaft. These are dendritic spines, and this blue structure maybe is a synaptic boudin or something. But these are individual synapses. This is wonderful. Well, there is a difference. There are a lot of differences between these two projects, but the main difference is that in the Human Blue Brain project, scientists are trying to understand both the structure and physiology of cortical microcircuits. But in the Connectomics project, scientists are just trying to map the structure of cortical connections. This is important. And even Dr. Lichtman mentioned this in one of his interviews, that they're just trying to understand the structure. And they hope that maybe because, well, of course, in physics and in biology, the structure of a complex or of a system has a lot to do with the function of that system. So maybe they're trying to actually understand or map the brain or cortical connectivity of the brain. And they hope that the structure, these maps of the structure or connectivity in the brain may tell them something about the physiology of them as well. But they're not doing anything about the physiology of these connections. But in the Human Blue Brain project, they're trying to understand the physiology of these different cell types of neurons and microcircuits as well. So that was it. That's done. So I just talked about the very important studies that are happening right now in the field of microcircuits. Of course, there are, I don't know, maybe thousands of other research projects, but none of them are as great as the Human Blue Brain project or Connectomics. None of them are as big as these two projects or studies on microcircuits. And well, in the next lecture, I'm just going to talk about the cell assemblies. I'm going to introduce cell assemblies because it is important. They are important. They are parts of a microcircuit. And I'm also going to talk about the dynamic aspects of microcircuits and how scientists are trying to understand the dynamics of a microcircuit, how a microcircuit and its neurons and their connectivity change over time. In this lecture, I just showed you, explain how scientists are trying to understand the real structural complexity of microcircuits. Thank you so much for watching. I hope you enjoyed this lecture. I hope you can watch other videos and lectures. And that's it. Goodbye.